Welcome to the cell-cell communication during development module. In this talk, we're going to learn about cell adhesion. We will begin with learning about morphogenesis and the main types of cells that are found in developing embryos. We will also learn about the main types of cell-cell communication, followed by the concept of selective affinity. We will then learn about the differential adhesion hypothesis. Morphogenesis is the process of arranging cells in such a way that they create organized structures and shapes. This process is very important for the development of organs and allowing these organs to function. The main types of cells that are present during development are epithelial and mesenchymal cells. The epithelial cells are able to adhere to one another, and so they're able to form complex structures like sheets and tubes. Mesenchymal cells, on the other hand, are loosely connected to one another, and more than often they are not connected to one another at all, which allows them to migrate. They're able to migrate to different places and are able to lay down the extracellular matrix. Now, organs are usually formed from epithelium and the underlying mesenchyme. During development, the cells have to have a way to interact and communicate with one another so that they can arrange themselves spatially and temporally in such a way that productive organs are formed as development progresses.
So now we're going to look at the basics of cell-cell communication. Now, cell-cell communication is mediated by proteins of a cell that can be secreted into the surrounding environment, or these proteins can be present on the cell membrane, or they can be inside a cell. Now, communication between cells can be local or they can be long range. And in this chapter, we're going to look at two specific types of cell communication, which are the juxtacrine types of communication, which results in juxtacrine signaling, and the paracrine types of communication, which result in paracrine signaling. So when we look at the figure to the right, and when we look at the figure that's part of the box, we're looking at juxtacrine signaling. And this is normally mediated by cell surface proteins that are present on two cells that interact with one another. When the same type of protein from one cell interacts with the same type of protein in another cell, it is called as homophilic binding, which is what we can see with the two yellow-colored membrane proteins on both cells. However, if you have one type of protein from one cell interacting with another type of membrane protein on another cell, then what we are seeing is called as heterophilic binding. During juxtacrine signaling, you can also have interactions between the cell and the extracellular matrix. Paracrine signaling, on the other hand, uses secreted molecules that can be called as paracrine factors. These are usually secreted from one cell, and these paracrine factors are sensed by another cell through the presence of cell surface receptors. We're going to see more details about juxtacrine and paracrine signaling as we go along the chapter. There are some terms that you must remember, and the first one is what is a signaling molecule, what is a ligand, what are receptors, and we already touched upon what is homophilic binding and heterophilic binding. Before we go into the whole process of cell signaling and different types of proteins involved in cell-cell communications, I thought we'd have a little touch-up on what are conformational changes. Now, we all know that all proteins are linear sequences of amino acids that are joined one after another. However, instead of being and existing as linear sequences, the proteins will fold to form a three-dimensional shape. So the conformation of a protein is the three-dimensional shape of the molecule. Now, proteins can undergo changes in these three-dimensional shapes or in these conformations when they bind to other molecules like ligands or second messengers, when they bind to other proteins, or when they undergo covalent modifications like phosphorylation, or ubiquitination. Now, signals from outside the cell can be relayed into the cell through conformational changes in the proteins. Many a times, these conformational changes provide a new function or property to the proteins. Now we're going to look at the concept of cell adhesion. So during development, the cells must migrate to their appropriate places to give rise to the different types of organs that are present in an organism. The sorting out of one cell type from another results from the expression of different proteins in the cell membrane, and we know that this happens through many different studies that have been performed in the past. So how did we actually find all of this out? And for this, we're going to look at one set of experiments that were performed by embryologists in amphibian embryos. Now, in the experiment, what the scientists did were that they took an embryo from one amphibian that had a certain kind of coloration in their cells, and they took a certain part of the embryo, which, say in this case, are the presumptive epidermal cells. So whatever was taken are cells that are going to say become the epiderm. They took another embryo where the cells looked different, and thus they have a way of differentiating which cells came from which embryo. But in the case of the second embryo, they took cells from the neural plate. What they did is, from these embryonic tissues, they dissociated all the cells so that they were all in single cell suspensions, and they mixed the cells from the different sources together. And then they waited to see what happened. Now what they found was that the cells that were dissociated from one another actually with time started to reaggregate, but it's in this ball-like structure or sphere-like structure that you can see in the blue box. But however, when we look at the type of cells and from where they came from, they were all uniformly mixed together. However, as time passed, there was segregation of different cell types. And so the cells that came from the epidermal part of an embryo were on the outside, whereas the cells that came from the neural plate were inside. So somehow these cells knew that they had to be placed in appropriate positions. So how does this actually happen? Now we're going to look at the concept of selective affinity that will help us understand how cells are able to migrate. So during development, the cells are able to sort out into the proper embryonic positions, and one of the ways that we are able to understand this process is by thinking that cells have a selective affinity for other types of cells and like to be with them, thereby sorting themselves to different positions in a developing embryo. For example, the inner surface of the ectoderm, which is a germ layer, has a positive affinity for another germ layer of cells, which is the mesodermal cells, but a negative affinity for the endoderm, which is the third germ layer. So what this basically tells us, the ectoderm likes the mesodermal cells but does not like to interact with the endodermal cells. Whereas when we look at the mesoderm cells, they have positive affinities or like to interact with both the ectodermal and the endodermal cells. Thus, the mesodermal cells like to arrange themselves in such a way that they're below the ectoderm but above the endoderm, or basically they like to be sandwiched between the ectoderm and the endodermal cells. What we have to remember is selective affinities can change during development. So we can have a certain population of cells that have an affinity for another population of cells at a certain stage of development, but as you progress during development, they no longer have that affinity. Now, many experiments were performed to show selective affinity, and that is what this entire figure shows us. So I'm going to go through some of the panels in order to understand what is happening in this figure. So when we look at the first column, basically shows that cells from the epidermis of a developing embryo were removed and dissociated from the embryonic tissues and mixed with cells from the mesoderm from an embryo, which were also dissociated. And in this case, what happens is, as we mix the cells together, we're able to see the cells are interacting with each other, but now there is a uniform distribution of both the epidermal cells, which are in blue, and the mesodermal cells, which are in red. However, with time, we see a migration of the mesodermal cells towards the center. And as development proceeds, we can see that the epidermis always stays on the outside, whereas the mesodermal cells are always enclosed but interacting with the epidermal cells on the inner part. Now let's do the same experiment but take mesodermal cells and endodermal cells. So they're dissociated from embryonic tissues and mixed together. In this case, the mesodermal cells are in red and the endodermal cells are in yellow. When we mix them together, they start aggregating with each other. And once again, we see more or less a uniform distribution of mesodermal and endodermal cells in the beginning. But as development progresses, in this case, now once again, we're seeing that the endodermal cells like to separate themselves from the mesodermal cells. If we take all three cell types, which is the epidermal, the mesodermal, and the endodermal cells, and mix them all together, all three cell types with time start separating from each other, and now we form different layers of the epidermal cells, the mesodermal cells, and the endodermal cells. So the cells know how to migrate and how to separate from one another during development, and this can be assigned due to the selective affinities that the cells have for each other and for other types of cells. We're now going to look at a hypothesis called the differential adhesion hypothesis. And what this hypothesis basically was built on the fact that during development, cells do not sort randomly, but actively move to create tissue organization. The embryologist Steinberg performed experiments that showed that certain cells migrate centrally when combined with certain cell types, but migrate peripherally when combined with other cell types. Now cells rearrange themselves into the most thermodynamically stable pattern. And hence this migration usually depends on the strength of adhesion between the cells. So when we have cell types that differ in the strengths of their adhesion that's present on the cell surface, that is when migration occurs. Cells that like to interact with each other more strongly or have a stronger adhesion tend to move to the center, while cells with the weaker adhesion tend to move outside. So if we look at two cell types A and B, and A has stronger adhesion than B, then A tends to move towards the center, while B tends to be in the periphery. On the other hand, if we take cells that are of type B and mix them with cells that are type C, and if B has a stronger adhesion to C, then in this case, B moves to the center while the C cells tend to be at the periphery. When we mix these cells together, what we can basically see, if you mix A cells and type C cells together, A cells have a stronger adhesion than C cells, and hence they will be at the center and the C cells will surround them at the periphery. The way these cells migrate basically depends on the strength of the adhesion that they have on their cell surface. So who determines the kind of adhesion that is present on the cell surface?
With this, we come to the end of our talk where we learned about morphogenesis and the different types of cells that are present during development, as well as the main types of cell-cell communication and the concept of selective affinity. We then learned about the differential adhesion hypothesis.